Okay, so today I'm going to be talking about meiotic recombination, the shuffling of DNA that occurs during meiosis, and the process by which parents transmit not one of their two chromosomes, but a mosaic of their two chromosomes to their offspring. And this process has been of longstanding interest to two communities, both of whom are represented at this meeting, but largely in parallel. So on the one hand, molecular biologists have been interested in mechanisms of recombination because in most sexually reproducing organisms, recombination plays critical roles both in aligning and leading to the proper segregation of chromosomes during meiosis. And in fact, in the absence of recombination, chromatids segregate randomly towards the poles, and usually aneuploidy ensues, a highly deleterious outcome. So in humans in particular, aneuploidy usually leads to spontaneous miscarriage, if not to severe developmental disabilities. So understanding mechanisms of recombination is fundamental to understanding of genetics and has important biomedical implications. But recombination also plays a role unrelated to meiosis per se in generating new combinations of alleles on which natural selection can then act. So in particular, the higher the recombination rate, the more rapidly beneficial combinations of alleles can be brought onto the same background or haplotype, and in general, the more efficient natural selection can be. And so evolutionary biologists have been interested in this process for just as long, but asking a very different set of questions, mainly centered around the conditions under which variants that modify the recombination rate, so-called recombination rate modifiers, will invade the population. In other words, if there are differences among individuals in rates and patterns of recombination, how do we expect them to play out over evolutionary time? And my interest has really been in trying to marry these two perspectives, in trying to use patterns, variation in recombination seen within and across species to gain insight into mechanisms of recombination and then hopefully integrate a better understanding of mechanisms of recombination into models for the evolution of recombination. Ultimately, what I'd like to understand is not just how recombination is specified across different species, but why it's specified in different ways in different species. What are the evolutionary implications of the way in which recombination is regulated, and how has natural selection shaped recombination mechanisms to lead to the diversity of recombination patterns that we see across the tree of life? And so we do that using model organisms as well as non-model organisms that are becoming increasingly accessible to at least some genetic analysis. So the story I want to tell you about today starts about 15 years ago with the discovery, mainly by sperm typing techniques, that humans, like most but not all sexually reproducing organisms, have recombination hotspots, which means that if you zoom into any particular region of the human genome, as shown here in a paper in an influential paper by Alec Jeffrey's group, then what you find is that recombination rates are very far from uniform in the genome. Instead, the double-strand breaks that lead to recombination events tend to be concentrated in regions of hundreds to thousands of base pairs, so-called recombination hotspots. And the discovery that humans had recombination hotspots suggested that if we wanted to characterize fine-scale recombination rates across the genome and learn about the determinants of double-strand breaks, we were going to need to get far below the resolution provided by pedigree analyses. And that posed a challenge because sperm typing techniques were not scalable, in fact, still are not scalable to the whole genome. And so it turned out that a method developed by population geneticists, originally to ask very different sets of questions, was really useful. So I'm just going to briefly describe the idea of this method. It's based on the fact that the patterns of genetic variation that we see today reflect events that occur in the ancestors of the sample. So if we were to sequence all the individuals in this room and focus on a particular region of the genome, if we then looked only at sites that differ among us, so variable sites, we might see something that looks like this picture. So each column here is a variable site where we carry blue or yellow alleles or both, and each row here is a haplotype, so each carry two of them. And so the patterns of genetic variation that you see reflect most trivially mutation events that occurred in our ancestors and led to these differences. They reflect population history and natural selection. They also reflect the recombination events that occurred from generation to generation ancestral to the sample. So if you were to consider two sites that are tightly linked, then the alleles at those sites will have been transmitted together from generation to generation and are quite likely to be found in the same haplotype in the present sample. And we see such examples where there's blue at one site and you're much more likely to see blue at another site than expected by chance. In contrast, if you focus on two sites that are separated, say, by a recombination hotspot, their alleles will be rapidly uncoupled, will be transmitted through different individuals, have different genealogical histories. There's no reason to expect them to be tightly coupled at present. So what that means is that we can ask the question, what recombination rates over the past are most likely to have produced the patterns of genetic variation that we see today? And what comes out the other end is an estimate of the so-called population recombination rate. So estimates that are time averaged over the generations ancestral to the sample and sex averaged over male and female ancestors. So another way to think about this is that although in a colloquial sense, all of the individuals in this room are unrelated, or at least most of us are unrelated, in a deeper sense, we're all related by some huge but unknown pedigree. And so what we can do is we can ask, what are the rates of recombination most likely to have generated the genetic diversity that we see in this room, summing over the possible pedigrees? And because the pedigree is huge and therefore includes a very large number of meioses, out of these kinds of methods, we actually get quite fine-scale resolution about recombination rates along the genome. So this type of method has now been applied to the genetic variation data that we have genome-wide in the context of projects like HapMap and 1,000 Genomes, and the position of over 30,000 hotspots has been inferred along the genome. And when this method really became feasible, applicable, we started thinking that an advantage that the method provided is that you don't need to bring species into the lab, you don't need to be able to cross them. Really, to apply this method, all you need to be able to do roughly, is to resequence a set of individuals. And so we thought we might use it to gain a first glimpse at what the evolution of recombination looks like at very fine scales. And we did this originally comparing humans to our closest living evolutionary relatives, chimpanzees. And this was in 2004 in collaboration with Svante Pääbo and Kelly Fraser, who collected 14 megabases of sequence variation data, so quite a big data set for the time. And using those data, we then asked whether the location of recombination hotspots in humans and in chimpanzees were coincident. And to our surprise, we found that not only were they in markedly dissimilar locations, but with the available data, we couldn't reject a model in which they were in fact independently distributed along the genomes of the two species. And we later confirmed that using genome-wide variation data from 10 chimpanzees. So that suggested that some aspect of the recombination machinery, potentially many aspects of the recombination machinery, differ between humans and chimpanzees. And it led us to ask the question of whether such differences might be observed even among humans. Might different humans use different sets of hotspots? And so to address this question, we used sort of a twist on a pedigree analysis that I don't have time to go into. But what we found is that indeed different individuals appear to be using different sets of recombination hotspots. So shown here is an example from a particular population sample in which each dot here represents an individual, and our best guess at what the proportion of recombination events that they lay down occur in a particular set of hotspots. So you can see there are individuals that are using that set of hotspots not at all, and individuals that are using them pretty much all the time. So most importantly for our purposes, because we were using pedigree analysis, we could show that this variation was heritable. That was an interesting finding in its own self, on its own, because here we have heritable variation for a trait that is fundamental and ensures proper segregation of chromosomes during meiosis. But it was also practically useful because, of course, once we find heritable variation for a trait, we can, in principle, map it. And indeed, mapping the source of this genetic variation turned out to provide a really important clue, revealing how it is that hotspots are specified, both in humans and, it turns out, in mammals more generally. So in a really nice example of convergence of lines of evidence from different disciplines of biology, in this case, mouse experimental crosses or mouse experimental genetics and evolutionary biology, there were 3 papers published back-to-back in 2010, all pointing to the critical role of a specific gene, PRDM9, in directing the location of double-strand breaks, both in humans and in mice. And this gene has essentially this cartoon, it has 3 main domains shown with this cartoon. The last domain is a zinc finger that binds DNA and is represented here in terms of the different fingers. So each finger is a rectangle, and the 4 letters in each rectangle are the 4 residues in contact with 4 base pairs of DNA. It also has a second domain, a set domain, that among other histone marks trimethylates H3K4. And the working model for how PRDM9 specifies the location of recombination hotspots in mammals is that it binds DNA facilitated by its binding motif, trimethylates H3K4, and then that mark, potentially in conjunction with other features, recruits a protein or a complex that then recruits SPO11. SPO11 comes, deliberately inflicts double-strand breaks on the genome, and then the repair of those double-strand breaks leads to crossovers and non-crossovers. So the version of PRDM9 carried by the human genome looks something like this. We can take the zinc finger and computationally predict what we would expect it to bind, and we find this position weight matrix that has considerable non-degeneracy. And so the binding affinity was since verified in vitro, but already the finding that recombination seemed to be directed by a protein with binding specificity was really interesting to an evolutionary biologist, because it raises the possibility that the hotspots that it specifies might be quite ephemeral. And the reason for that has to do with the details of how recombination occurs. So when a break occurs on one of the two homologs, that homolog is not the donor of information in the repair process, but the recipient. In other words, if you envisage two alleles, a blue allele and a red allele, if the break occurs on the blue allele, the repair process will lead to two red alleles. If the break occurs on the red allele, it'll end up with two blue alleles. So at the population level, if red and blue are equally likely to experience a break, nothing changes. Half the time you have two red, half the time you have two blue. But if instead of red and blue, these are hot and cold alleles, alleles more or less likely to recruit PRDM9, then by definition, breaks are more likely to occur on one background than the other. And so what happens is that you actually see overtransmission of the cold allele. And that, at the population level, leads to the loss of the hot allele. So in other words, if you imagine a set of hotspots shown in this cartoon, and a cold allele were to arise at one of these hotspots, we would predict that it would very rapidly invade the population, leading to the extinction of that hotspot. And there's essentially no countervailing force because individual hotspots contribute very little to alignment and segregation. So you can lose the hotspots one by this process of overtransmission of the cold allele, and that poses a conundrum that Rosie Redfield termed the hotspot paradox on the basis originally of observations in yeast, which is if hotspots lead to their own extinction, why are there hotspots at all in natural populations? And so a possible solution to this paradox, in mammals at least, is provided by changes in trans to the binding domain of PRDM9. Notably, if the zinc finger of PRDM9 mutates to recognize another sequence, then it can restore a whole new set of hotspots. And one could even imagine that there's quite strong selection for that restoration process, because while marginally there's nothing to prevent the loss of individual hotspots, eventually those are going to imperil proper alignment and segregation, and so there might actually be selection to restore hotspots. So consistent or supportive of this scheme for hotspot evolution, we know that there's rapid evolution of the fine-scale recombination landscape. We had shown it in primates. It was subsequently shown in rodents. We know as well that there's preferential loss of hot alleles on the human lineage. That was shown by Simon Myers and colleagues. And finally, we know that the zinc finger of PRDM9 is indeed rapidly evolving. In fact, it is the most rapidly evolving zinc finger in the human genome. So what I'm showing you here is just that zinc finger represented, again, in that same cartoon format in different mammalian species. And you can see that not only are the residues that are in contact with DNA different across species, but the very number of fingers differs across species. And importantly, this rapid evolution of the zinc finger is not due to some inherent mutability of the sequence, because the rapid changes are focused at those residues in contact with DNA. So this is really selection for new binding affinity. So all these different lines of evidence make quite clear that PRDM9 plays critical roles in directing recombination in the genome. Where does it direct recombination? Well, in humans, it seems to direct recombination at some distance away from the transcription start site. So what I'm showing you here is a plot where every transcription start site in the human genome was collated at position 0, and then I'm plotting the average recombination rate as a function of distance from the nearest transcription start site. And you can see there's a tendency for recombination to occur tens to hundreds of kb away from the transcription start site. So this is seen in humans, is also seen in chimpanzees, and it's seen very clearly in mice. So these are results not from my group, but from Dan Camarino-Otero and Galina Petukova's group. So they took an experimental approach in which they could get a quantitative readout of double-strand breaks in the genome, and they asked the question of where those breaks are occurring in a wild-type mouse first. And by their operational definition, pretty much none of those breaks were occurring at the transcription start site. They then engineered a knockout mouse for PRDM9 and re-asked the same question, and interestingly, they found a much larger fraction of breaks at the transcription start site. So their interpretation for this finding is that in the absence of the H3K4 trimethylation marks laid down by PRDM9, the recombination machinery is somehow defaulting to the residual marks that are present in the genome, which tend to occur at or near promoters. And consistent with the pattern in knockout mice, when Adam Boyko and Adam Otten took a population genetic approach to study dogs, which are of interest because they carry a pseudogene for PRDM9, they found a pattern that was very similar, where recombination was concentrated at the transcription start site, and intriguingly specifically at transcription start sites very close to CPG islands. So motivated by these observations, we were interested in studying a natural population that lacks PRDM9, not a knockout mouse, not a domesticated species, but a natural population that doesn't carry the ortholog to PRDM9 to verify that indeed recombination was occurring at the transcription start site, and most of interest to us to elucidate what the implications were for the evolution of hotspots and whether hotspots still evolved rapidly. So in doing so, we were guided by a hint that had been provided by Chris Ponting's group before we knew, in fact, what the function of PRDM9 was in recombination, and that is that the chicken genome didn't seem to carry an ortholog, potentially raising the possibility that it was lacking from birds. And so we verified that using the genome sequences made available by the Bird Consortium and also using RNA-seq data. And indeed, we do not find an intact ortholog of PRDM9 in birds or in crocodiles, although we do find one in turtles, and I'll return to that at the end. So guided by this observation, we decided to focus on a specific taxon of birds, Australian grassland finches, and so we focused on zebra finch and their close relative long-tail finch and an outgroup double-barred finch. And we chose this group for a number of reasons, but in particular because the nucleotide divergence among them, the extent of genetic differentiation, is almost identical to what is seen between humans, chimpanzees, and gorillas. So it provides us with a nice comparison to a timescale over which we know that in mammals there's rapid evolution of recombination hotspots. So then we sequenced roughly 20 of each, long-tail finch and zebra finch, in an outgroup sequence. We inferred, we called genetic variants, we inferred fine-scale genetic maps. And what I'm showing you here is essentially a sanity check. On the y-axis are our results based on the breakdown of allelic associations. And on the x-axis are results that come from a pedigree analysis of a couple hundred crosses from Hans Elgren's group. And so at the scale of megabases, which is the scale of resolution of the pedigrees, you can see that our results are highly concordant. So reassured by this kind of sanity check, we then proceeded to ask where recombination is occurring in the bird genome. And consistent with what had been found in knockout mice and in dogs, we see that it is concentrated at or near the transcription start site. So we can refine this a little bit more, and we can ask whether it's specifically concentrated at transcription start sites near CPG islands, and the answer to that is yes. And then we can ask the converse. We can say, conditional on being at a CPG island, does the distance to the transcription start site matter? And the answer to that seems to be barely. So there are a number of possible interpretations for this finding. One is that the recombination machinery is actually targeting both H3K4 trimethylation marks and CPG islands. Another is that it's not those genomic features per se that matter, but rather that they denote sites of transcription initiation where nucleosomes are displaced and where the recombination machinery simply has more access to DNA. The next question we asked is whether birds have hotspots, the answer to which isn't obvious. Not all recombining species have hotspots. In particular, Drosophila are not thought to have this kind of hotspot. But the answer for birds appears to be yes. We find them throughout the bird genome. And then most of interest to us, do hotspots evolve rapidly? So to ask that question, we went back to the genome-wide data that we had collected in 2012 for chimpanzees, and we asked what fraction of hotspots overlap between humans and chimpanzees by some sensible operational definition. And then we asked, well, what fraction would we expect to overlap by chance, given that hotspots have to be somewhere in the genome? And so to do that, we placed the inferred hotspots at random along the genome, or independently in the genomes of the two species, but mindful of the broader scale recombination rates. And what we found is that even this really simple model for the extent of overlap expected was sufficient to basically recapitulate what we observed. So observed is in blue here, and what we would expect by chance is in orange. So barely more hotspot sharing than expected under this naive model. We then asked the exact same question of the long-tail finch and zebra finch, and I'll remind you that they have very similar levels of genetic differentiation. But here we see a completely different pattern, where when we expect only about 3% sharing by chance, we see over 70% of hotspots shared between the two species. And because simulations suggested that we should detect very few spurious hotspots shared between species, but of course we don't have 100% power, we think that this is an underestimate, and in fact, most, if not all, hotspots are shared between the two species. So at this point, we sought corroboration for what we were seeing, and we took advantage of the fact that hotspots actually have two completely independent effects on patterns of genetic variation. On the one hand, they break down allelic associations. That's the signal we had used to infer their location in the first place. But they also increase GC content. And they do so through this process of GC bias gene conversion, which is that when a double-strand break occurs and heteroduplex is formed, if that heteroduplex involves an A or a T on one strand and a G or a C on the other, then it's preferentially repaired to GC. And that process acts actually in a way that's mathematically analogous to selection for GC content. So it leads to excess GC substitutions in regions of high recombination. And so what we did is we said, well, if we look at those positions in which we saw allelic breakdown and called hotspots, do we also see excess GC substitutions? And we did so in hotspots relative to cold spots that were matched for the same overall base composition. And so the answer is yes, and you can see that here in hotspots and cold spots for zebra finch and longtail finch, and GC star is just a measure of excess GC substitutions. So seeing that was highly reassuring, and it also gave us an idea, which is that if we can see this footprint of hotspots in excess GC substitutions, then maybe we don't need to sequence 20 whole genomes and call genetic variants and look at allelic associations in order to learn about hotspots. Maybe we can look at those same positions in more distantly related species of which we only have a single genome and see whether we're seeing the footprints in the same location. And so we did that, and indeed, we see this footprint of hotspots extending over tens of millions of years all the way out to collared flycatcher. So circumstantially, at least, suggesting conservation not just over millions of years, but tens of millions of years in birds. So to recap, in species that carry PRDM9, such as apes, as well as mice, we see this tendency for recombination to be moved away from the transcription start site. In contrast, in finches, in knockout mice, in dogs, as well as in yeast species that lack PRDM9 and in fact lack binding specificity directing recombination, we see recombination concentrated at or near promoters. And these taxa also differ dramatically in their rates of evolution. So in apes that carry PRDM9, as well as in rodents, there's essentially no sharing of recombination hotspot locations. In contrast, in the finches that we studied that do not carry PRDM9, we see this extensive conservation of hotspots. And in fact, published back-to-back with our results on finches came a paper by Scott Keeney's group that used an experimental approach to look at a very similar question in two really diverged yeast species, and similarly found that they shared not only hotspot locations, but heats, despite very high levels of divergence. So together, these observations suggest a link between how recombination is specified in the genome and how it evolves, where when recombination is directed by PRDM9, hotspots can be rapidly lost through overtransmission of cold alleles, and they can be lost and gained through changes to the binding specificity. In contrast, when recombination targets functional genomic elements such as CPG islands, or more generally sites of transcription initiation, then we see conservation of hotspots, presumably because in that case, those hot alleles cannot be lost because they play important pleiotropic functions. They have other roles, notably as sites of initiation of transcription. And so, of course, the question is then, is this really general? Is it inevitable that if you have binding specificity, you have rapid turnover of hotspots? If you don't have binding specificity, there's stasis. And so we're starting to get at this question, and as a first step, we reconstructed the phylogenetic history of PRDM9 invertebrates. So that's what I'm showing you here. To the left is the phylogeny. To the right is a cartoon of the gene structure. And so the first thing to note is that we see an intact ortholog of the mammalian PRDM9 that we know to be playing a role in recombination arising somewhere near the origin of tetrapods, either before or after the split with amphibians. if we hypothesize that the intact ortholog is required in order to have PRDM9 directing recombination, we would predict that, for instance, that other vertebrates, for instance, fish, should behave like PRDM9 knockouts. And that we have now a number of lines of evidence suggesting that indeed they do, despite the fact that they carry a fragmentary ortholog to PRDM9. Their genomic patterns look like they don't carry PRDM9, at least in terms of its role in directing recombination. So here's one example. We built a genetic map based on ancestry switches in naturally occurring hybrids of two swordtail fish species. And essentially, the ancestry switches are informative about where crossovers occurred. And what I'm showing you below is the same kind of plot of the proportion of recombination events that occur at a given distance from a transcription start site. And you can see that they show this pattern in common with, for example, the knockout mice and finches. We can try to further refine which tetrapods actually use recombination to use PRDM9 to direct recombination. By recalling that where PRDM9 directs recombination, we predict that its zinc finger should evolve very rapidly. And so what we do is we essentially test for evidence of rapid evolution of the bindings of the residues in contact with DNA using an approach that was introduced by Oliver et al. Or Wrinkle et al. On that approach. And so what we find is evidence that some species, at least, of snakes, turtles, marsupials, as well, of course, as placental mammals, show evidence for positive selection in the zinc finger. And interestingly, those are exactly those taxa that carry the intact ortholog to PRDM9. So on this basis, we would predict that the crab domain and SSXRD domain, about which much less is known, probably do play also important roles in potentially recruiting SPO11. Also, we would predict on the basis of this that snakes and turtles and marsupials should show rapid evolution of the recombination landscape, and we're starting to test that now, starting with snakes. So in summary, what we and others have shown is that vertebrates employ at least one of two strategies to specify meiotic recombination. They either do so by targeting recombination to promoter-like features, or by using PRDM9 to specify the location of recombination hotspots. And so knowing this, we can recast the sort of general question of why different mechanisms of recombination in terms of a question about the emergence of PRDM9, and in particular, why it is that it acquired this role in directing recombination. What was beneficial about PRDM9? Sufficiently beneficial that we see it conserved across mammals, and potentially it looks like across tetrapods, and yet not essential because we see it lost time and time again in birds and crocodiles, in canids, in amphibians. So one hypothesis actually is a hypothesis that has been around evolutionary biology for a very long time, which is that maybe, or at least some form of which has been around evolutionary biology for a long time, which is that maybe there's an indirect benefit that stems precisely from the role of PRDM9 in moving recombination away from transcription start sites. So in particular, if you imagine that there are a lot of interacting regulatory and coding alleles, you may not want to uncouple them. And so a recombination rate modifier that moves a large fraction of recombination rates in the genome away from the transcription start site could potentially be favored. Another possibility, which is not mutually exclusive, is that PRDM9 is a weapon deployed in the arms race that all these species are engaged in against selfish genetic elements. And so hints pointing to that are that, in particular, in a number of species, we see the binding, or the predicted binding site of PRDM9 enriched in a subset of transposable elements. And another hint is that we've inferred at least the origin of this new role of PRDM9 to be the origin of tetrapods, which is concomitant with a time where we see this huge expansion of crab zinc finger proteins from an ancestral PRDM9, and we know, of course, that an important function of crab zinc finger proteins is in silencing transposable elements. So we're currently testing these hypotheses. I'm going to leave you on the open question of why it is that PRDM9 was favored, why it is that animals switch strategies, and I want to thank a number of people who have contributed to different components of this. So Graham Coop, with whom I had a close collaboration analyzing the human genetic variation data. I glossed over that, but it was very complicated, and it was wonderful to work with him on it. Gil McVean, with whom we analyzed the genome-wide chimpanzee data, and his former postdoc, Adam Otten. And then past and present members of my lab, Ellen Leffler and Sonal Singhal, who led the bird data analysis, Zach Baker, who did the phylogenetic analysis and is now becoming an expert of boa and sea turtle recombination, which I don't think is exactly what he signed up for when joining my lab, and Molly Schumer, who brought the swordtail fish model to the lab. Thank you.
Thank you so much. All right, so our very last speaker this evening is David Kingsley from Stanford University, and David also works at that